Hi, in this lecture, we're going to talk about the central nervous system and specifically an overview of it and then talk about how it's protected. So you probably remember that your central nervous system is your brain and your spinal cord, and the two work together as basically the control centers. And so what they do is the information comes into the CNS. Remember, you have your sensory or afferent information, bringing information in. And then the CNS is the control system. So it integrates, it processes, it makes decisions, and then it sends its instructions out through that motor or efferent out to its effectors. So when we look at the brain, we're going to start with that. This is just some trivia. You don't have to write any of this down, but I think it's kind of interesting if you think about your brain controls everything, right? But it's mostly water. This is why it's so important to stay hydrated. There's about 100 billion neurons in the brain, which is, they say, is about the same number of stars in our entire galaxy. So if you've ever gone outside on a crisp, clear night and looked at all the stars, right? Energy, if we could hook it up, could power a 25 watt bulb. And they say the average number of thoughts is about 70,000 thoughts a day. And think about your thoughts, right? They're not always positive thoughts and those tapes that we're constantly telling ourselves. If we look at the basic structure of the brain, we know that the brain contains what's called CSF, which is cerebrospinal fluid. So this is the fluid inside the brain, and it's inside chambers that are going to be called ventricles that we'll look at soon. Now, you've heard of ventricles, I'm sure, in your heart, but you also have these hollow spaces in your brain as well. If we look at the brain, if you kind of think of it like a giant mushroom where you have the stalk of the mushroom, the whole cap part is the cerebrum. So the cerebrum is the largest part of the brain. It's kind of the top part of it. If we were to go below, so we're going from superior to inferior, in here, this blue structure is called the diencephalon. Now, encephalo means brain, and the diencephalon is going to contain things known as your thalamus, which you may have heard of that before, and your hypothalamus. Just abbreviate. We'll be talking about all of these in more detail. We have what's called our cerebellum. And the cerebellum is that smaller lobe kind of on the bottom of the brain. I always think it looks kind of like cauliflower. So your cerebellum helps with posture and balance. And then we have what's called the brain stem, which is this area down here. You can see why it's considered almost a stem. And the brain stem is going to be made up of something called your midbrain, something called the pons, and the medulla oblongata. And you can think of it like a stem because it kind of forms the bulk of the stem of the mushroom. And this controls a lot of our vital functions, right? It's going to control reflexes and heart rate and breathing, etc. Now, if we look here at a section of the brain, this is a frontal section to the brain, you can also see these little spaces in here. These are the ventricles I was talking about that are going to contain the cerebrospinal fluid. And we also know that the brain is going to have gray matter and white matter. Now, let's look at the spinal cord real quick. We have a whole lecture just on the cord, but we know that the spinal cord is going to begin way up here at the medulla oblongata. So the medulla oblongata is the most inferior portion of the brainstem, and the spinal cord joins with it. And where they meet is at that big hole in the base of your skull called the foramen magnum. We know that inside the spinal cord, there's a little canal right in the middle of it. It's called the central canal, and that also contains cerebrospinal fluid. So CSF is found in ventricles inside the brain. And you'll see soon that there's 4 ventricles in the brain, and it's found inside the spinal cord in what's called the central canal. Now, we also have white matter and gray matter, and you probably remember that white matter is white because it has so much of that myelin on it, and the gray matter is gray because it's unmyelinated, and it's also all the cell bodies of the neurons. Well, if we look at the brain and the spinal cord, notice that the white matter in the brain is deep. See how it's deeper? And the gray matter is superficial. But it's opposite in the spinal cord. In the spinal cord, notice the white matter is superficial and the gray matter is deep. It just kind of flips a little bit. Now, you've learned some terms we used to talk about this type of structures, and one is called a tract. And a tract is a bundle of myelinated axons. So it's a bundle of axons in the CNS. We call that a tract, and it will look white because of all the myelin. When we look at the gray matter has all those cell bodies, and so we have what are called nuclei and centers. And we're going to just combine them as the same thing. A nuclei and a center are a cluster of cell bodies in the CNS that have a related function. And we usually call them centers. So there's a cardiac center in your medulla, and you probably guess it controls heart rate. So some other terms to add to your list of terms. All right, so obviously these structures are critical. So what is going to protect them? Well, our main protection is obviously our bone, right? We learned the bones. We have these connective tissue coverings called meninges. We'll look at the cerebrospinal fluid, and then the brain has an additional protection called the blood-brain barrier. So bone, right, you have a skull, very hard, protects your brain, and you have your vertebral column. And remember that you looked at the vertebral column and you saw that foramen, that hole where the spinal cord passed. So bone's pretty important, basically forms a helmet for your brain. Now, the meninges are connective tissue coverings. They're actually pretty strong and tough, really strong layers. And there's three. So there's the dura mater, the arachnoid, and the pia mater. Now, the dura mater is the outermost one. So we're labeling these from superficial to deep. So you can see the dura mater out here. So I'll put it, that's for superficial. And then you can see the arachnoid is the one that kind of looks like a spider web. See this in here? It looks kind of web-like. And then the pia mater is the deepest one, and it actually attaches directly to the brain. So when you look at a brain, and we'll have some sheep brains for you to check out, you'll see this kind of shiny covering on it. That's the pia mater. So it's the innermost meninx, and it actually attaches pretty tightly to the entire brain. Now, the middle one is called the arachnid or arachnoid. And since it's in the middle, there's going to be a couple spaces associated with this one. So you can see the membrane looks kind of like web-like, arachnid, spider web. So you can see the layer here. Now, you have your arachnid, so I'll just put the A here. And then you have your dura mater. I'll color this purple here. Okay. The dura mater out here, and then remember you have your pia mater, which is attached directly to the brain. So there's a few spaces that we need to define. There's something called a subdural space and a subarachnoid space. Now, the subdural space is the space that's between the dura mater, remember this is the outer meninx, and the arachnoid. So we come over to this picture now. It looks a little different. Let me go over a few things. So we have the dura mater, and you're going to see soon that the dura has two layers. So there's a layer here, and there's a layer here. And you're going to see this space in between. That's called a sinus. A sinus is just a space. It's called a dural sinus. And they have names. We'll deal with those shortly. But I'm going to color this blue. So I have my dura mater, and then I'll just kind of do this purple thing for my arachnoid. And you will see a little space here, which is my subdural space. So that subdural space is the actual space between the dura, which I'm coloring blue, and the arachnoid. The subarachnoid space is the space that's between the arachnoid and the pia mater. So again, we see our arachnoid is purple, and I have my pia mater, and then I'm going to have my subarachnoid space, which is where a lot of this web-like stuff is. Now, it's the subarachnoid space that has cerebrospinal fluid, and that space is a little bit bigger than the subdural space. So the subarachnoid space is filled with cerebrospinal fluid. It also has the larger blood vessels that are coming up to the brain, and it has these things called arachnoid villi. And this is a villa. See how it kind of sticks out? This is the arachnoid, and it sticks out into the sinus. So it has these arachnoid villi, and these are really important because this is where CSF is going to be drained. So you have a note here about a lumbar puncture or a spinal tap, and if that's done, they have to take a needle and go into the subarachnoid space. Now, they don't do that in your brain. They do that in your spinal cord, down around L5 in your lumbar region of your cord. Now, another space we have is an epidural space. So we have a slightly different view here, and we're coming back to the dura mater. So the dura mater is the outermost meninx. The name of it, dura mater, means tough mother. It's very, very strong. And it does have 2 layers to it. So again, in this picture, we can see the dura mater. So I'll try to color that as like a blue color here. Okay. And then remember that it's 2 layers. So I'm just going to color this whole thing blue. And then remember the space in between is called a sinus. And again, we're going to deal with that very soon. So I have my dura, and then underneath it, right, my arachnoid and my subarachnoid space is probably what you see the best there. And then I'll just go ahead and try to draw the pia here. See how the pia actually attaches directly to the brain? So when we look at the dura mater, it has 2 layers to it. And there's something called an epidural space. So the epidural space is the small space that's basically between the bone, right? Because you're going to have your bone and the dura mater. So you'll have you might see it better in this picture. Let me come back here. So you see your cranium, right? You have your bone, and then the epidural is going to be a space between the dura mater and the bone. So epi means above, right? And sub, when we're looking at subdural, that means below. So that might help you figure out where these are. Now, we do have these spaces, and we have these blood vessels that are bringing blood to the brain. So sometimes we can have bleeding into these spaces, and that's what a hematoma is. So a hematoma is excessive bleeding, some kind of trauma, usually some sort of brain injury or head injury, is going to cause a blood vessel to break, and you can have a subdural hemorrhage or an epidural hemorrhage, and you have a couple pictures here of these. So epidural is out, it's more superficial, right? Epidural is between the dura mater and the skull, where subdural is going to be in basically that space between the arachnoid and the dura mater, and you can see your subdural hematoma here. So they're both of concern. They can both put pressure on the brain and need to be treated pretty quickly. Now, when I was showing you the dura mater on the previous pictures, you saw it was 2 layers. I'll go back to the blue here. So we have let me try that again. We have the dura mater, and we have 2 layers to it. So you obviously can see your bone, and then you have the dura mater, okay? Now we know that the dura mater is the outermost meninx, but it also kind of goes inside into the brain a little bit in these like invaginations, and it forms these folds. And between these folds are usually these spaces, which are called sinuses or dural sinuses. Now, notice that you have the arachnoid villi. See them there? These are those finger-like projections. Remember that they're draining the cerebrospinal fluid. The cerebrospinal fluid is here in the subarachnoid space where you see all the web-like stuff. So the CSF has to be drained all the time. And those arachnoid villi are draining that cerebrospinal fluid into the dural sinus, because the dural sinus is collecting all that cerebrospinal fluid, and it's collecting venous blood and returning it back through the veins that are going to collect all this blood. Now, the dura mater actually folds. It actually forms these sinuses by going inside the brain in a couple areas. So I'm just giving you their names. Don't worry too much about memorizing, but this is where we have sinuses in the brain. We'll see some down here. We'll see 1 here, and we'll have 2 here. So these folds kind of go in the brain and separate the cerebrum. So you can see the superior sagittal sinus and inferior go right down the middle, right, with one of those folds. You have a fold that comes in and kind of forms like a floor of the brain, and one all the way down by the cerebellum. And so these are where the dura mater comes in. It has a separation, which is called a sinus, which is a collecting area for blood and cerebrospinal fluid. Now, if we get infection or inflammation, there's 2 things that can happen. We can have it in our meninges, right? Meningitis or encephalitis. Now remember, encephalo means brain. So encephalitis is going to be more severe than meningitis, but they can both be pretty bad. So meningitis, inflammation of the meninges, it can be caused by many different types of things. A lot of times meningitis can be spread through saliva. So you might hear about meningitis outbreaks at daycare and college campuses, right? We have lots of saliva mixing. It can have, the symptoms can be a sudden fever, headache, stiff neck. They usually do blood work, and a lot of times they'll probably do a lumbar puncture just to see what kind of organism is causing it. A lot of times it's bacterial, so it can be treated with antibiotics. Encephalitis is worse because it's an infection of the brain is inflamed. A lot of times it's from viruses. You'll hear about these viruses being spread through mosquitoes. Mosquitoes feed and pick up, like there's a Eastern equine encephalitis. So because the mosquito feeds on the blood, it can pick up the virus, and then when it feeds on a person, spread it that way. But again, this could be very, very bad. It could be fatal. And so a lot of times they need to do some hospitalization when you have an encephalitis condition. Okay, cerebrospinal fluid. This is the fluid remember, it's inside your brain. It's inside your spinal cord, so it's within the brain and the ventricles, but it's also surrounding both the brain and spinal cord and that subarachnoid space. So your brain floats in it, right? There's a little bit all the way around the brain, all the way around the spinal cord, so it's a cushion for your brain and your cord. So if your head moves quickly, the brain is sloshing around in there, but it has that fluid around there to help cushion. Now, in addition to cushioning the brain, it actually helps to transport nutrients and chemicals and waste products. So it helps transport materials with the interstitial fluid moving back and forth. Now, CSF, by the way, should be clear. It's a clear fluid because it comes from your plasma. And there's structures called a choroid plexus, and they have cells called ependymal cells, which you saw in the neuroglia lecture. And so these are found in every ventricle. And these are the cells that make and circulate the CSF. Remember, the ependymal cells have the little cilia that can help circulate it as well. So CSF is formed from ependymal cells, which are in clusters in each choroid plexus, and they line the ventricles to help circulate the cerebrospinal fluid. Now, it does come from plasma, but it has fewer proteins. Plasma has a lot of protein in it. They can't get inside that. And so a concern of this is that proteins are really good buffers. So cerebrospinal fluid isn't as good at buffering materials. So what you're seeing here is a bunch of those ependymal cells with the cilia in a choroid plexus. And again, its job is to filter blood. So the blood leaves and goes in the interstitial fluid, and it forms cerebrospinal fluid, which then circulates. And as it circulates, it helps remove waste and drop off nutrients. Now, it's made at a continuous rate. So it's always, formed and drained at the same rate. Remember, the arachnoid villi drain it into those dural sinuses that we looked at. And those dural sinuses, remember, are collecting CSF and blood, and they're sending it back out of the brain. You might have heard of your internal jugular veins, right? They're going to collect all that and bring it back to your heart. So if it accumulates, though, it could be a problem. So you can see in this image the brain, and if the ventricles become enlarged, again, you're increasing intracranial pressure. So that's a concern, with the pressure building up and compressing those neurons. So hydrocephalus happens in babies. And what happens is, for them, luckily, the skull can expand because they have fontanelles. So as that fluid increases, the skull does to basically accommodate that increase. So usually they can figure out the cause of it and maybe put a shunt in or a drain to help drain that fluid so it doesn't accumulate. If this happens in an adult, once you have sutures, now that's life-threatening because your skull can no longer expand. So it's going to cause an increase in pressure. And a lot of times they'll do a little craniotomy. They might even remove part of the bone a little bit to give room for that brain swelling. And then, as we mentioned earlier, it is used for diagnostic purposes. So a lot of times you do what's called a lumbar puncture. And remember, you need to go into that subarachnoid space so that you can get a sample of the fluid, and then you can examine it to find out maybe if it was meningitis, if it's a virus or a bacteria. So we said the CSF circulates around the outside, and it's also found in the ventricles inside the brain. And there's 4 ventricles. 2 of them are called lateral ventricles, and they're kind of like C or comma-shaped structures, and they're inside each cerebral hemisphere. So we haven't talked much about the brain, but we said the cerebrum's on top, and there's a right and a left cerebral hemisphere. Well, inside those hemispheres are these spaces called lateral ventricles. Well, there's a membrane that separates them called the septum pellucidum. Septum needs wall. So that's the membrane that separates the 2 ventricles. And there's a little hole, interventricular, right? The hole between the ventricles that moves the CSF back and forth between them, because the CSF is going to circulate. It's not stagnant. It's going to flow through the ventricles. From the lateral ventricles, it flows into the third ventricle, which is right in the center of your head, right inside the thalamus, which was part of that diencephalon we just said. From the thalamus, it flows to the fourth ventricle. Well, the way it gets there is this little channel called the cerebral aqueduct. Now, if you want to call it mesencephalic aqueduct, knock yourself out. Cerebral is probably easier to remember and spell. From the fourth ventricle, it then flows into the central canal of the spinal cord. So we have this flow of cerebrospinal fluid, and in this view, you can see the lateral ventricle. See how they're kind of C or comma shaped? And then you have a lateral view here. So it's formed and it circulates through the lateral ventricles down into the third ventricle, through the cerebral aqueduct to the fourth ventricle, which is right behind the pons, which you'll be learning shortly. And from the fourth ventricle, it goes down into the central canal of the spinal cord. And here's another view that really is helpful, I think, at seeing the flow of CSF. So again, we have these choroid plexes that make it, and it's flowing from my lateral ventricle to my third, down the cerebral aqueduct, and you'll see this on the brain models. This is the fourth ventricle. It kind of looks like a triangular shaped structure, and then from there it goes down into the central canal. Remember, it's also flowing around both the brain and the spinal cord in the subarachnoid space. And then it's all going to be drained finally by those arachnoid villi into the dural sinus. So it's constantly made, it circulates, and it's drained. And you have a brand new batch about every 3 to 4 hours. And I read somewhere that's only about 150 mLs, which isn't a lot of fluid, but it's enough for your brain to float in it and to protect it. Now, the blood-brain barrier, just what it sounds like, is a protection only associated with the brain. And so what we're seeing here is our astrocytes, which you learned about previously, and we're seeing our blood in a capillary. So what this is a physical layer of astrocytes. So you have these cells, and what the cells are doing is wrapping themselves around the blood, okay, and also between the blood and then a neuron. And so anything that's trying to leave your blood and get out has to pass through this green cell first. So your neuron would be up here. So it's a layer of cells. Now remember, astrocytes are the most abundant neuroglial cell. They form the blood-brain barrier, and their cell membrane is a bunch of phospholipids. So what that means is only anything that's lipid soluble can pass through this cell to get to your neuron, which means oxygen and CO2, because they're very, very small, lipids, or anything lipid soluble, is able to leave your blood, pass through the astrocyte, and get to the neuron. Anything that's charged or big, like hydrogen ions, cannot pass through the blood-brain barrier. This is important because if you're trying to treat any brain disorder, if the medication is big or charged, you can't deliver it through the blood. You'd have to put it directly into the brain. So this is a problem when we're looking at some of our treatments for brain disorders when we're trying to get a drug to get into the brain. For example, Parkinson's disease is caused by a decrease in dopamine. You can't just give dopamine because it can't get through the blood-brain barrier. So they give them a precursor. It's called L-DOPA, because L-DOPA can pass through those astrocytes. It can pass through the blood-brain barrier, and then the brain uses it to make dopamine. So that blood-brain barrier is pretty important for protecting the brain. And if we just look at what some of the main blood vessels are going to the brain, the main arteries are called the internal carotid artery and the vertebral artery. So the internal carotid artery, here's the common carotid coming up the neck, and then it separates and it comes all the way up into the brain. And there's the vertebral artery. It actually comes up through those transverse foramen and the cervical vertebra. And on our cervical spine, our C-spine models, you can see these arteries coming up. And just note that there are some areas of your brain where your brain releases big hormones. And in those areas only, the blood-brain barrier is more permeable. So those hormones can leave the neurons and go into the blood to travel. Now, because we have these blood vessels coming to the brain, we need to talk briefly about injuries to these blood vessels. You know, we talked about hematomas earlier and how they can be problematic. You can also have cerebrovascular accidents, and that's a fancy name for a stroke. Now, if we were to have a stroke occur, these are just some common signs and symptoms. You don't have to memorize them all. This is more like for you to understand, right, that how they start. Numbness, vision problems, confusion. You want to get checked out immediately. But I do want you to know there's two types of strokes. There's a hemorrhagic stroke and a ischemic stroke. A hemorrhagic stroke is I can't talk today, a brain bleed. It's like usually an aneurysm. A ruptured blood vessel causes bleeding to occur. I'm going to move myself up here. Whereas ischemic is due to a blood clot. They have very different treatments. So usually there's stroke teams that are going to be at the hospital. They're going to do an MRI or a CT scan because you have to know what kind of stroke it is. The faster you can stop the bleed or treat the clot, the less repercussions your patient's going to have. So if it's ischemic, they're going to give a clot buster. They're going to try to dissolve that blood clot. If it's hemorrhagic, they got to stop the bleed. So they might go in surgically and put devices in to clip or coil to basically stop that bleed from happening. This is why it's so critical that you know what kind of stroke it is. Because if it's a hemorrhagic stroke, you don't want to give a clot buster, right? A blood thinner. They'll just bleed out. So cerebrovascular accidents are strokes. They can come out of nowhere, and you can see some common signs and symptoms. And if you experience them, you do want to get checked out very quickly so you can get the proper treatment. So this concludes our first lecture talking about the brain and spinal cord and the protection for it, or for both.